Thank you. Good afternoon. Welcome back. Last panel session with lots of expectations, I would say. And you know now all panelists, because everybody was either speaker or panelist, so there is no need for special introduction, except we have Dr. Ken Kono from Japan, from National Institute, National Institute of Sciences, NIH in Japan. They are responsible for biopharmaceuticals and cell and gene therapies, and they are already doing a great job, so we'll hear more about that. And we have another colleague from Japan, from NIID, National Institute for Infectious Diseases, Dr. Koji Ishii, who should be online. Hi, Koji. And they are responsible for vaccines. So we will hear from two different perspectives in terms of NGS application in Japan. And we also have Dr. Aimir Ramkishan from the National Regulatory Authority in India. Hello, thank you for joining. And I think that we should also have one regulator from Nigeria virtually connected. Maybe if Igisha is not yet with us, she will be with us soon. So we are waiting for another picture on the screen. And we also have several other regulators from other countries also connected and listening with attention. So we can actually start exploring the situation in different countries. We started yesterday. You remember that we discussed what is the situation in Indonesia, in Bangladesh, and in Egypt. So we will now fly to Delhi. So Dr. Ramkishan, would you like to share with us what is the situation in India, given that you have quite a large number of manufacturers, Indian biologicals, particularly vaccines, have been pre-qualified by WHO. And in that context, how do you see these scientific advances coming along with the evaluation of biologicals in future? Are you as a regulator ready to go along with this, or would you like to share with us what are the challenges and what do you need as a technical support from not necessarily this group, but maybe from many collaborators who are willing to help WHO to support you? Over to you.
Good evening, Dr. Ivana. Thank you very much for inviting me here to participate as a panelist. And all esteemed panelists, good evening to one and all. I'm very happy to interact with all eminent people sitting on the dais and off the dais. And yes, as far as Indian regulations are concerned, we are having well-defined guidelines in our Indian legislation with respect to high throughput sequencing and next generation sequencing methods with respect to the biological products. And Dr. Ivana yesterday gave the presentation with respect to different countries' perspective, consisting of India and Korea, Japan, South Africa, and 25% people are having some challenges with respect to sequencing as an alternative to existing in vitro and in vivo advantageous virus detection methods for viral safety, testing activities of different biological products for their marketing authorization or clinical trials for different products. So this is the thing. And as far as India is concerned, yes, we are having provision with respect to promoting this high throughput sequencing methods. And our guidelines are very clear with respect to the Indian law that we are having well-defined Schedule M that deals with good manufacturing practices, and Part 4 has given specific requirements for manufacturing of biological products as per the provisions of Drugs and Cosmetic Act, and also national guidelines published by Indian Council of Medical Research in the year 2017 is very clear with respect to the cell and gene therapy product development and clinical trials are concerned. So, and also Indian government published in 2019 GSR 227E, which was published on 19th March 2019, is also very, very clear with respect to this important high throughput sequencing as an alternative with respect to carrying out this advantageous detection methods for the viral safety. So this is a thing, and we are very, very clear to enforce. And second thing, we are following WHO TRS guidelines and also ICH guidelines as well as 993 and 998 is also very clear 2010 and 2013 guidelines, because as far as India is concerned, being a vaccine above the biological products and other important biotherapeutics, we are not compromising and putting efforts to take care of all important viral vaccine safety measures are concerned. The only thing is we need to have special things, so validation of the important methods and lack of some expertise with regard to the validation of these HPS method, and also some of the cell lines and other raw materials also when we go for the master cell line and all, and process related parameters and manufacturing operations and control. Then again, related to products also, we are taking lead with respect to provisions of Indian law as far as this advantageous testing is concerned to ensure high quality immunobiological product. But we need to have some focus with regard to how the European pharmacopoeia is taking care with respect to this HDS method with respect to that. So we need to have our Indian pharmacopoeia also need to have special focus for using this test for the next generation sequencing method. And as a regulator, we are also having discussion to accept in toto with next generation testing as an alternative to other testing of advantage agents. And also we want to replace all RRR, refinement, replacement, reduction. So we need to replace all the existing in vivo model to in vitro testing methodologies. So these are all the things. And with the support of WHO, under the supervision and dynamic leadership of Dr. Ivana, scientist and technical team head with respect to biological, and as a ECBS member, we can take lead globally to harmonize with all your esteemed panelists today. And India is leading with regard to promotion of all immunobiological products to promote international health. So this is my views, and we are all here to accept. And only thing is we need to have well-defined protocol to understand for the validation methods and protocol to sense that, so that in near future, definitely we are going to take lead. But this is one of the expensive method. We need to have educative approach and create awareness how the global standards are developed and how the reference materials are being taken care with the support of NIBC UK. And all our expert committee members are going to take lead with respect to these important testing methodologies for promoting and also ensuring high quality immunobiological products, particularly vaccines and and other biological products like biotherapeutics and biosimilar products. Thank you very much, madam, for giving me the opportunity. We are very clear. Only thing is we need to have capacity building as per the WHO guidelines and also World Health Assembly resolution passed in 2014, 67.15 is also very, very clear. We need to have the access, availability, and affordable biological products for the global community, but at the same time, academia, regulators, and stakeholders and all vaccine manufacturers and biological product manufacturers also to take lead. And this is the one of the challenge. This interface would be geared up with the eminent people like you under the supervision of Dr. Ivana. We can take lead. Thank you very much for giving me the opportunity to share some of your thoughts with respect to high throughput sequencing alternative model for replacing all in vivo methods. Thank you very much. Thank you very much.
Thank you very much, Dr. Ramkishan. The audio probably was not very clear, but I assume that a majority of people understood that there is a willingness in Indian NRA, and it seems that in terms of the guidance and also following international guidance, everything is in place, so they are prepared to review this data. The question is really in terms of practical arrangements, whether you are receiving this data from manufacturers. We also have Indian manufacturers here in the room that they may wish to comment on that as well. Or may wish to reveal the secret when and how they are going to submit their high throughput sequencing data together with submission for licensing vaccines or other biologicals. But it's good to hear that you are prepared for that. We also heard that you are prepared to go ahead with the replacement of in vivo testing models, the current models. So it will be interesting to hear where are you in terms of the replacement of in vivo with in vitro tests?
So,
is this still work in progress, or do you think that?
So we are in the process, and proactively we are going to take lead with respect to replacement of all these in vivo methods. Only thing is, the challenge is we need to have well-defined and validated methods. If it is available, definitely our government is committed to take lead with respect to effective implementation of these important in vivo tests towards focusing on in vitro model testing on biologics. And companies also constituted, and for cell and gene therapy products, Indian Council of Medical Research, and under the supervision of Dr. Raghuvanshi, Director General, Drugs Control General India, we are also having CBBTDC, Cell-Based Therapeutic Drug Evaluation Committee, with consisting of all experts from different disciplines and consisting of few experienced regulators, those who are having command on biological products, taking lead and having consultation and following the international guidelines, especially on this thing, WHO TRS guidelines. Take care.
Thank you. So basically waiting for validating model methods. So maybe this is something that we will discuss further, and will be interesting also to hear if there are suggestions on how to move forward. Maybe we can now fly to Tokyo to hear Dr. Koji Ishii. What is the situation in Japan? I presented yesterday your feedback saying that Japanese manufacturers did not submit HTS data to you, but we know that regulation in Japan is a bit complex because you are working on vaccine evaluation. NIH colleagues are responsible for biotherapeutics and cell and gene therapy products. PMDA is the overall body for as a national regulatory authority. So would you like to elaborate what is the situation and how do you see the way forward in Japan? Because we expect that Japanese manufacturers or maybe other manufacturers will come soon to you. So are you ready for that?
Yes, thank you very much for inviting me to this meeting. And I would like to talk about the situation of vaccines and quality control in Japan. There are several live virus vaccines in Japan, such as MMR or smallpox. But these vaccines have producing a long history, more than 50 years, mainly produced by domestic companies. And they first did the adventitious virus detection by using in vivo test, and then after that using in vitro cell culture test. But they are, I think they are satisfied with this, the result of the in vitro testing, and they are a little bit reluctant to change to the new sensitive high throughput sequencing method. So we have to persuade these domestic companies to introduce this new high throughput sequencing method instead of in vitro cell culture system, because this cell culture has many disadvantages. I think yesterday several presentations said that only the virus which can replicate in the testing cells can be detected. So high throughput sequencing is much better method for the adventitious virus detection. So I think we have to collaborate with domestic companies to introduce these methods for the detection. I think this is the situation of Japan.
Thank you very much. Dr. Koji Ishii, do you want to comment on that in terms of?
As Imana mentioned yesterday in your presentation, yes, I agree that Japan is a little bit conservative in introducing the new method to review for the review of products, medicinal products. But as Q5A was revised, and the PMDA, the Japanese regulatory authorities, is interested in the new technology, NGS, especially in the NGS. And I frequently I communicate with PMDA officer often, and they are very interested in my NGS data. And yeah, and yes, so they are waiting for the products in which the NGS was performed in replacement with in vivo test. I think they are willing to consult, yeah, the review there.
Thank you very much. I don't know whether this is surprising to the audience and panel members to hear what is the situation in Japan. I was surprised because I expected, as Japan was one of the founding members of ICH, I thought they would be the first to introduce this in their practice. But in fact, it takes time, and we understand also the practical issues and the need for discussion with manufacturers, and also this kind of regulatory preparedness also to be in charge of this review, to develop the expertise, to be able to provide advice. Because in this case, when manufacturers are running really fast and going quite a long way, building the expertise, regulators have a difficult position to step in at the stage when their knowledge and experience may not be at the same level where the manufacturers are. This is not the first situation that we are seeing. It usually industry is actually moving along with new technology. So innovation always goes like that. We saw it many times before. But in terms of regulatory preparedness, I think it will be important to hear what people think. How can we help moving forward? Obviously, all these IBS events that you heard about yesterday and today help moving HTS from research tool now to let's say, regulatory tool, but it still requires additional steps in building the expertise, in developing this trust actually in this innovative tool, if you wish. And also phasing out animal testing is quite challenging task, both for manufacturers and for regulators. And I think it's also important what our colleagues from India and Japan mentioned, the role of pharmacopoeia. We heard that European pharmacopoeia is revising the chapters. Chinese pharmacopoeia is also working on that. I understood that Indian pharmacopoeia is also interested to become a kind of proactive in this field. So it's really important to hear in practical terms, what are the next steps? What is most needed? Because we cannot provide all tools and assistance at the same time, but maybe if people would like to share their thoughts and views on 1, 2, 3, what we can do, because it's end of 2024. In coming 2 to 3 years, what should be the most important step to be made in order to move forward? And I see that Laurent has an idea.
Yeah, no, I just wanted to comment that, of course, ICHQ5A has a defined scope, and the live vaccines and inactivated vaccines are excluded from the scope. However, I think the principles which are described are applicable to all biological products. What I want to say also is that the European Pharmacopoeia chapter is also applicable to all biological products. It has been drafted for that purpose. And also, as we mentioned with Gwenaël yesterday, the chapter 2616 and 523, they are for both vaccines, viral vaccines, but also for gene therapy products. So I think there is no barriers regarding the applicability of these HTS, NGS methods. They are really, I will say, applicable to all these products. And I hope that the publication of the final version of the European Pharmacopoeia chapter describing both the technology and the validation guidelines will support the implementation, because I think it's a way to not standardize it, but at least have an approach for validation which is described. And I remember in the past, it was the same for at the European Pharmacopoeia level for the PCR for mycoplasma, for example. When the text was published, it has also been a boost for the use of the technology. So I hope it will be the same.
Any other thoughts or questions?
No, I think really in promoting the excellence, that's what comes from my discussions with other regulators. Of course, we are in charge in assessing the new methodology, and always everyone is hesitant if he gets to assess something that he doesn't understand. So I think in order to promote, we really need to have dissemination amongst the regulators and also have a good training and good agreement what needs to be checked and have some standards. I think the Pharmacopoeia is the first way forwards. Of course, the difficulty is really that the rapidly evolving field, and even when we are trying to write this chapter, we have to stay still flexible because it's such an evolving technology. But really here to get an assessment and get some training or some to assure the regulators and making them understand about the advantages of the technology, but also giving them some guidance how to assess, or at least the cornerstones and the key factors that should be assessed. I think this might promote, or I hope it could promote, the acceptance.
I just wanted to further that thought in terms of all of the various spiking studies that you heard about during this conference and the list that I presented. And I know there are others that, people are doing, and hopefully the completion of these studies and the publications, one of the goals is an effort to develop SOPs for the different types of matrices and and what not to do and what steps and how to optimize. So it's moving towards that in terms of, trying to have some general SOPs that could be used. So we hope that, there'll be additional, sharing of knowledge and efforts to, that will help towards that. That seems to be needed as well, in addition to the training or a part of it.
I forgot to mention, there are also the case studies from the ICH implementation working group that could also help. I don't know if you want to comment, Marie, or...
Thanks, Laurent. And I know that Johannes has mentioned this as well yesterday in the regulatory presentation with respect to the scope of the Q5A is obviously quite large, for the revision, but the introduction of NGS is a very, very significant component of the overall revision. And since its publication, we have been working in the implementation working group in developing training material for really key updates within the guideline, prior knowledge, continuous manufacturing, and the introduction of the new ATMPs, viral vectors and viral vector-derived products. But in that training material, which is prepared for dissemination from basically from the start of next year, what we have is integrated the application of next-generation sequencing throughout the case studies. So we're giving direct examples of new product types as well as conventional monoclonal antibody production, where next-generation sequencing is being used in these examples as a direct replacement or a supplementation, depending on the examples, for the conventional assays. So it really is, we've forwarded the, I suppose, the application of NGS now within these examples as well. And this training material is ready for dissemination per ICH. Yep.
That's very helpful. Actually, we are facing here two issues. Well, not only two, more than two, but two in terms of building capacity in the world. If we want to see global acceptability of HTS as innovative tool, there is a lot that needs to be built actually in the world. And starting with the expertise, which is always taking a lot of time and efforts, and sometimes the whole professional life spent in building the expertise in one area is not even enough to reach the stage of the up-to-date scientific advances. So we see this as a particular challenge in the countries that have not actually started working on that yet, while, you know, in the other part of the world, there are already 15 to 20 years of experience. So this is not easy to match, and this is something that we would like to explore, how we can speed up that process. In terms of the expertise, we heard very good examples of collaborative efforts in the teams that have virologists, that have bioinformaticians. This bioinformatic expertise may not be present everywhere in the world. So just to maybe put expectations into realistic frame, from WHO perspective, we are thinking first about the kind of discussion with regulators in so-called manufacturing countries, where regulators are exposed to quite a large number of producers. So for instance, India and China are two very good examples, Indonesia as well. Producers are bringing this innovation to regulators and are kind of pushing for further developments. So if those who have already been in this journey for a long time would like to share what are the useful starting points and how to move this forward. I agree that there is a lot already available, meeting reports from previous discussions, training materials. There was a webinar organized by IABS in September. There was a discussion with developing country vaccine manufacturers. But one really has to go through all these, and usually regulators from countries that WHO is working with are asking for help to summarize this, to translate this into case studies or review of examples, to make it a little bit, you know, user-friendly, rather than just leaving everybody with a long list of available sources. So any suggestion on that point, which is more like, you know, how to move forward with educational tools, and probably identifying what needs to be developed in addition, knowing what is already available.
I guess one obvious, you know, that I can just mention, and I think you've been hearing about this through the whole conference, is the just, you know, anyone is able to join this Advanced Virus Detection Working Group. It's a very interactive working group. Membership is open as long as, you know, it's not for, I mean, it's a scientific exchange, and very, you know, it has a lot of experts there. So in terms of sharing knowledge, and, you know, you're free to ask questions, bring new topics to the table that might be discussed further. So I think as a first step, you know, I would invite, you know, those who are not members and who want to learn more about NGS to join the group. And as you've heard, you know, we've got experts and discussions pretty much on the different steps in the workflow of the NGS in the different subgroups, and the subgroups themselves, they're led by experts, and we have a range of participants, early users, people that haven't, you know, started NGS, and the experts. So it also provides an opportunity, you know, for contacts and for knowing, you know, who has what expertise, who's doing what. And I think, you know, that will at least provide you with, you know, trying to understand the current status of where we are. And then a lot of times we do spend time going, you know, sort of reviewing, you know, where we were, where we are now, how we got here. Sometimes we do devote efforts, you know, meetings to that. So I think that could be a helpful, and the meeting is, you know, it's 2 hours every other month. So, you don't have to spend too much time, and you might learn, more than you expect.
Thank you. Other suggestions?
This forum is fantastic because it's bringing the regulators together. And the first question we're asked many times is, has the methods been accepted by the regulators? First question, very often the only question. So if there's an answer to no, or we don't know, it doesn't go very far. I see this very much as a journey. It's probably a long journey, but it is still a journey that we have to take people on. In my past history, I have been working very closely with regulatory authorities, providing training specifically on a one-to-one basis, closed-door meetings. These were incredibly beneficial, and they were very much around just classical standard biosafety testing. There is an assumption that everybody knows and understands why we perform the tests that we do. They don't, and that was the key point. If we can maybe turn that on its head and start doing something similar for next generation sequencing. There might need to be something more practical, because I don't think it's that easy to actually understand from the point of view of explaining in kind of theoretical terms. It might need to be practical, and I'm open to questions or suggestions of how we do that. But I think that would be really helpful to make sure that when the regulators are really familiar, but actually even the companies are more familiar with what the technology can achieve and what are the kind of the challenges and the support that is available for that to work. I did a review recently, and I had ended up writing like a 50-slide document of all history of NGS, which is incredibly detailed, and all the publications. If you go back and read everything, it'll take you quite a long time. So if you maybe summarize some of that into a kind of very specific training deck, and perhaps the training decks that are going to be from the ICH Q5A will help. We can maybe base that information on that particular type of analysis. I think it's a start, and I think it's what is really needed, because not everybody can attend these forums. So it would be nice if we can take this out a little bit further.
Yeah, that's a good suggestion. And also, I think all the materials or most of the materials shared in this conference will be publicly available. So I think we are trying hard to make as much as possible available. But in practical terms, I think it's also important to look from the perspective of the access to biologicals of assured quality. And in that definition of biologicals of assured quality, regulators are playing a very important role, actually key role, because they are responsible for review of these products. And when they are dealing with the licensing or overview of clinical trials, we have very precise parameters in WHO benchmarking tool, and regulators are also assessed and classified in terms of maturity level and all these. So for those who are not familiar with that, this is also an opportunity to introduce this innovation through this system, although it takes quite a lot of time. The question is that if regulators are actually in their regular job reviewing the data and they're not satisfied with what they are seeing for a variety of reasons, and they ask for additional data. And we heard yesterday the example of influenza vaccine, which, by the way, the secret was revealed. It's coming to Japan pretty soon, I understood, right? So the question is that in that review, regulators may ask for additional data, and we always recommend early dialogue between regulators and manufacturers, always recommended to manufacturers to go and discuss with regulators before submitting the dossiers. And we heard yesterday that there was actually also lesson learned in the example of influenza vaccine. That may save a lot of time, because in the end, it is in the interest of all involved to have access to biologicals of assured quality, safety, and efficacy. So one of the potential problems and challenges will be delay in completing evaluation in case that there is no expertise, there is no experience. Regulators are willing to review this, but they don't have expertise and they are not prepared for that. Are there some efforts that manufacturers are planning to do in, overcoming this potential gap and maybe avoiding delay of several months or year in the review of these data, or maybe there are lessons learned that people are willing to share. Silence means
Maybe this means we should
means
move on
not
with
willing to share or
Well,
not
no.
facing this challenge.
This might be a good time maybe to go into the general discussion topics that we created. And I think some of those points may come up again, if that's okay. Because we want to be sure that we cover that also in the meeting. But it's been great, the great discussion and very good points that came up.
Just maybe one thing to add to your points, Ivana, there is the reliance aspect, which is also important between regulators. So I don't know if you want to comment on that, but I think it's an important point.
Yeah, this is a great concept, but is getting very, very slow into practice for a variety of reasons that we can discuss maybe as part of this discussion.
Yes, thank you. Yes, so yeah, we have prepared a list of questions to also bring the panel up to date and to see if you can also comment, because we have covered a number of topics during this conference, but maybe there are additional ongoing efforts that are related to NGS implementation. So I don't know if one of you or several of you want to comment on that regarding your own activity that we did not cover maybe during this conference. Do you have something that you want to add regarding the additional efforts, ongoing efforts, maybe in fields that we did not cover completely, maybe in the cell and gene therapy or other domains where you see that there are progress which are ongoing that we did not discuss? The panel or from the audience, by the way.
So I think we are doing NGS routinely. So for the first point, it's not a question of technique. So I think the third point is interesting. We have already discussed this point regarding viromics and genomics. There are now standards, viral standards, which are widely used. No? It's a success. Now cell lines for the transcriptomic activity are still lacking. And I think it's not really an issue, but it could be very useful to have such standard and begin to compare the different pipelines based on the same type of material. Yes.
So actually, we do have a study that is being initiated to evaluate the virus detection from the infected cell lines with different levels of expression. And to make it more complex, we're also adding the virus panel to it. So it will mimic a complex matrix. It's in the background of a unprocessed bulk. So that's like our next study following the Nimble first study that Allison presented. So we are looking into that. And then additionally, the effort is to then, based on that result, to open it up for a collaborative study to then evaluate the cell lines more broadly with different laboratories and pipelines. So then we'll announce that when we're ready to have that study after we've got some initial results using the cells ourselves in our own study. And additionally, I also wanted to raise the question, since we moved to standards, that we were very much interested in knowing more about the kind of challenges that are being faced in having the access to the current model viruses, because that would also help us as we prepare the cell lines as potential model cell standards, so that we can at least look forward or thinking forward, so that we can address these challenges that we've heard a little bit about, the, I guess, issues in terms of, it's not the availability, it's the accessibility. So we'd very much like to hear, and also from the colleagues that are online, what can be done to make it more accessible. And it's not clear to me exactly where the challenges are. Some may be regional, maybe possibly political, I don't know, but we can try to address them if we hear your thoughts about what can be done, and then we can try to see if maybe the virus stocks that we currently have, they can be provided in a different repository as well, that may be more accessible, but we need to know where should we, yeah, so I see Sigrid has some thoughts about that. Great. She may have had challenges.
So maybe just to add, because we asked for the viruses, we are based in Europe, so it needs to take a flight. And so in Belgium, it needs to come to a particular airport. Otherwise, they are destroyed and you need a permit. And so the carrier that wanted to do it was not delivering to that airport. And so then all the problems started. So I think if there would be stocks available in Europe, that it doesn't need to come by flight, that would already be, for us, a big help, because we had it twice, the issues, and it took 3 or 4 months to get everything solved.
Well, I'll let Blandine talk for. Yeah, is there a repository that can store them and distribute them free of charge? Or okay, in Europe, or well, if there's some charge, then I guess you guys will be willing to pay for it. So, but, or which country should that be in? I mean, like I said, it's up to you guys to let us know, and then we can provide it, send it over there. So
maybe I can clarify that for all WHO reference materials, it's always with WHO custodian laboratories. So we are not asking volunteers here. Don't take it wrong. It has to be in one of WHO custodian laboratory. And we will probably ask our colleagues in MHRA in UK, because they have a lot of experience. They're distributing 99% of WHO standards all over the world, have a lot of experience, and they know how to do that. So it has to be done properly. Otherwise, people will not receive this in a good shape, which is needed.
So just to add, because of something I'd heard, will there be any issues for accessibility if it's in the UK?
Yeah, I think
That's always the case.
Yeah, no, that's true. I think it would be nice if we could have a stocks in Europe, but from my point of view, the UK is not the easiest, because now we need more and more documents for UK. But in Europe, another place could be nice.
But I would say even having it in an EU country will not solve all the problem, because we have to ship it outside in the world. And I think when we established at the Paul-Ehrlich the Zika virus standard, we finally had to decisions to make it non-infectious. I think really sending out infectious cultured substances, there are so tight regulation. You are in additional being asked on the animal components and the veterinary. It's always a nightmare. So I think just transferring the stocks elsewhere will not solve all the problems, I'm afraid. And just to expand this, with the Zika virus, we had to take the hard decisions. Okay, we have to inactivate it, knowing that probably the heat inactivated Zika that we are distributing is not the intact virus particle. It's clear. And then we have to look carefully on the stability of the heat treated stuff and so on. It's complicated, but it's a kind of compromise with accessibility and preserving it. It's much more easy. It was much easier with the blood-derived standards, having clinical samples with the native virus from blood. But whenever it comes to cell-cultured virus, it gets very difficult with the regulations.
So that's good to know. So these are non-pathogenic viruses. They're BSL-2 viruses. And additionally, they're characterized in terms of, and by NGS, what's in the vial. So would that still cause a problem?
Yeah.
So we tried to brought in very infected cells from US to Europe. It was a nightmare. 6 months.
So Vero cells are a different.
Yeah, but it's with animal-derived material. We should not think it's easier than human material or human infectious material. It can be worse, and it depends really on the countries, etc. And I think that is a good discussion because we need to consider when we develop the next standards, cell therapy, cell-infected cells. Is it reasonable to do that? Should we find other solution standards like RNA transcripts to make it a practical, feasible way? It's not perfect, but then we don't have any distribution issues.
So again, this was trying to make it more close to real life in terms of, spiking cells in uninfected cells, so you know what your limit of detection is. But just to get back to the question about the animal-derived materials, so even if it's purified virus, so these have been purified, they have a certificate of analysis from ATCC that go with it. They don't have bovine components. It's been because of the purification. I think.
No, certainly, there are two questions. The one is whether the material contains animal-derived material, such as serum, but the other things are the veterinary regulations. So even sending a totally non-pathogenic PCV1, you have to adhere to the veterinary regulation, which might also be tightly. We have to have clear in Germany documentation where we get it, what we are doing it, when we dispose it, and we have to. So the issue is really sending infectious material that has been propagated by cell culture.
So PCV1 actually became an issue with the USDA in the US also, but we addressed that and they cleared it because it's non-pathogenic. So at least in the US we were able to deal with it, but maybe not in Europe. So
I need to add it again with the Vero stuff. We did it twice. Yeah, and it's specific. We know that.
Yeah.
But just to let you know, and it's our experience, and I guess Merck has the same experience. We have different sites in US, in Europe, and we ship material on a regular base, right, to do testing, what cannot be provided at one site, etc. So it's a nightmare. I can tell you, it's really delays everything. And if that can be somehow avoided, it's really what we should. And I think really it's something what we need to consider when we're developing those standards, etc. Unfortunately, it sometimes depends on a person. It takes 3 days to get it through custom. Everything is fine because all paperwork was provided upfront. And the next time it's destroyed. Don't tell me what the reason was, right? Because the paperwork was the same. So that is what is really happening.
There are obviously challenges with distribution, and it's a very good point that we should think in the preparation of the next standard. But I think for the actual distribution of this standard, it's a good idea to have at least 2 points of distribution rather than only one.
no,
That's something that we
that's
can.
feasible, but I'm hearing that it's not going to solve the problem.
Maybe
Maybe
To certain extent,
Okay.
it will,
Right.
because for most of European destinations, it will be fine. What is difficult to resolve if it's required by Iranian laboratory or national authority, or if it's required by Russian authority or laboratory, that these are difficult destinations for which, again, through WHO regional offices, we sometimes have some kind of assistance and solution. But you're right, it's not 100% feasibility. But having European distribution center will definitely reduce a lot of problems for majority of.
we can more definitely work on that to move towards that for sure. So maybe we can go to, you already skipped down to 3, so maybe we can go to 4 then. So I guess what additional efforts are needed for establishing or implementing NGS? And I think this is sort of extension of Ivana's discussion earlier. And so now we're opening it up to everyone in the room, as well as the folks online as well. And I guess first the panelists. Okay. Yes. So what are the additional efforts? We already heard about the reference standard issue. So what additional efforts are needed for establishing NGS?
Well, honestly, we were talking a little bit about the reliance model and the breaks, and I actually would like to hear more about that. So, what I think it's been, correct me if I'm wrong, fairly successful with COVID, although that was obviously there was an urgent need for that in terms of distribution and getting other NRAs to understand that those vaccines, et cetera. But yeah, it seems maybe for the audience here, can you explain a little bit about a WHO reliance model, how it's been successful and what the challenges you're facing now? Because I think understanding that would be very helpful to the audience. Yeah.
Yeah, actually, there are quite a lot of experience, and I think people in the room, as far as I know, are familiar with this. But maybe we can just mention that in a number of areas, but particularly in the area of vaccines, a lot of benefits were gained through the reliance model, and there are also collaborative efforts in the networks. And we have several regional networks where regulators in the same region actually work together. So for instance, in Africa, there is a network of regulators that are working together. There are also inter-country groups that are helping each other. So for instance, if one regulatory authority is more advanced than the other, they are then helping. And this reliance actually started with this kind of building trust. So working together, 4 or 5 regulatory authorities in sub-region, if you wish, build this kind of concept where 3 or 4 in neighbor countries or relatively close countries can rely on the decision made by one regulatory authority, or at least they gain the experience with the assessment that was actually conducted for the evaluation of vaccines or other biologicals. So this is how it works, that they have actually sort of joint review, or there is also parallel review when 2 regulatory authorities are reviewing this in parallel and they are not necessarily in the same region or sub-region. We also had a help where, for instance, Health Canada was helping regulators in developing countries. We had also other regulatory authorities being very much involved. I think CBER also provided help to regulators in several countries. So there are different models on how it works. The reason why I mentioned that it's sometimes slow process is because not every regulatory authority is willing to go for the reliance concept completely. So there are sometimes kind of borderlines where people would like to keep their independent assessment. And we are always emphasizing that from WHO perspective, it is the full right of the National Regulatory Authority to make their own decision. They can actually use the experience and assessment conducted by another regulatory authority, but the final decision is a decision of the regulatory authority in charge. And this is also the same with WHO pre-qualification. Pre-qualification is the system that provides evaluation for regulatory authority that does not have sufficient expertise or experience, but the final decision is always made by the authority. And we had the cases, of course, in majority of cases, they accept WHO pre-qualification assessment, but we had also cases where regulatory authority said, no, that's not enough for us. We would like to have more kind of assurance, and we would like to review the data by ourselves.
Yeah, sorry. I guess I should have clarified a little bit. I guess what I was asking, well, thank you for that. That is helpful. But I guess I'm asking, have you explored using that reliance model for NGS implementation in particular? And what has been the response of maybe, as you said, some NRAs are less receptive to that model? I guess, you know, you don't need to name names or anything, but have you looked at that? What is the, you know, are there challenges with that? What are the thoughts with regards specifically to NGS implementation? And would this reliance model work for that to help NRAs that are less experienced with this technology?
No, actually, it was not explored just for this purpose, because reliance goes beyond that. So it's not just reliance in one aspect, it's actually reliance in the entire assessment. But I think when we continue with our survey, we just started actually exploring what is the situation in different countries, but we'll definitely go into that, and not only through the questionnaire, but really in the discussion with regulatory authorities, because it's very important to understand what are the expectations and the needs in each regulatory authority. And as I mentioned, maybe starting with regulators in manufacturing, in producing countries, because they are much more exposed to the actual questions and review of the data that are submitted to them. Reliance is the concept that can actually work for the others, but we guess that for those in producing countries, they would probably prefer to have this expertise and experience in their hands to be able to evaluate data generated by their local manufacturers. And local manufacturer today means also global manufacturers. You can imagine that, you know, there are no borders as it used to be before.
Yes, actually, I thought I would just mention when it came to the topic of the vero cells. So as you know, the vero cells, CBER is one of the, I guess, holders of part of the vero cells at the ATCC repository. I'm the project officer. However, so, I mean, when we get a request, I send it over to ATCC, and then somehow they've been shipping it all over the world, and it's a black box, and they've never complained to me about the shipping. So maybe, we can call on an ATCC representative here, Heather, who's been my contact for a long time, to maybe, reveal the secret of how are you shipping these vero live cells all over the world, including China. And I mean, I know we get requests from everywhere. Yeah.
Hello. We just prepare by getting our CITES permits. So ATCC ships all kinds of cells everywhere all the time. So we always have all the permits for all the countries. And when they expire, we get a new permit. For the WHO cells in particular, we have to get a new permit every time because each of them has its own batch number. So they each have to have their own permit, but we just go through that process.
And I should mention that the 5 viruses, the original model viruses, we had a contract with ATCC to store and distribute them, and they were being distributed. So I hadn't heard of this problem till now, so again, I mean, maybe they can share their experience as to how you overcame the hurdles in the different regions. But we did, I know some were challenging and 2 shipments came back, but I think that was more because of the courier issues in some of the countries, which we've now addressed. But, I mean, it seems like it hasn't been such a challenge, but maybe now the restrictions are different.
For the most part, it's communication, I think, is the issue. So in most cases, we get all the information from where we're shipping to, and we know how to get it there. Sometimes we have even had help from WHO to get things places that we need to get them. Not for this, but for other things. So I mean, we generally know how to get things where they're supposed to go. So in some cases, for example, this Belgium case, they may not have known it had to go to a particular airport, or at least our courier obviously didn't know. So that, we need that information up front. So I would just say, if there is anything like that and you're asking for the viruses, then make sure you share it in case somebody doesn't know.
I have just maybe a comment on coming back to the reliance topic. Not really the reliance itself. We have a network of batch release laboratories in Europe, which is coordinated by EDQM, and we are, of course, doing testing where you have mutual recognition, and there is only one national control laboratory, OMCL, as we call them, who is doing the test for the others. So it has been very successful during the COVID-19 pandemic, and because the certificate which was issued was recognized by many authorities, even outside of our network, in fact, because it was a pandemic situation and there was a need to have access to these vaccines very quickly. And our procedure was really transparent regarding the tests we were doing and all the batch release process. I just want to say that it's what we see with the OMCL network in Europe is that it's difficult to have all the technologies. So sometimes we have some specialized centers and we are promoting that. It's one of our strategic axes from the OMCL network strategy, to promote to have some specialized centers on some technologies, because it's difficult to maintain and to acquire this technology. And I think it's a little bit the same with NGS, and to have worked on that in the past for the implementation. The bar is quite high, and we have to acknowledge that and to consider that maybe having some specialized centers in different regions, of course, would be a good solution as well for having this technology in place. Probably not all NRAs need to have the technology. They need to have the expertise to evaluate the dossier, as mentioned by Arifa, but maybe not all have to have the hands-on expertise and the technology internally, because it's a very challenging step.
And speaking on that, we recently had a discussion, if we zoom all the way out, just about the terminology. Do the regulators understand if we say NGS and they are saying HTS? And I know that it is actually because we just had the talk, so I looked in the ICHQ5A where it is mentioned immediately. And that was our solution also internally for our documentation when we have audits, if it says NGS and they are like, but it's called HTS. So I don't know if that is a problem for anyone, if they have had that issue or anything.
And maybe just to comment on that, I think within the ICHQ5A guideline, we had this exact same debate. There's been a lot of surveys over and over, and we were very, very clear within the glossary of the guideline that the equivocation of the terms are right there. So there should be no ambiguity, because we know that there's preferences in different areas.
It also says it in the chapter immediately that it is interchangeable.
Yeah, and likewise in the draft pharmacopoeia chapter 2641, we had also exactly the same discussion with the group of experts. So we call it high throughput sequencing, but I think in the first paragraph also you have an explanation that this is also known as a next generation sequencing. So there is no ambiguity because we are aware that there are different acronyms that are used in different documents. But I think there is a link, so
I think that is good then. But I was just curious if anybody had faced an issue, because we talked about, do we need to change all our documentation?
I don't think it will be the problem, because in most of the documents, it is actually mentioned as interchangeable terminology. In WHO document, we are using HTS. And the reason for that was that our expert committee on biological standardization advised us not to use the term next generation sequencing, because they mentioned what will be then the terminology when the next generation of next generation comes into place. So next generation sequencing was considered as a kind of research tool and a little bit kind of promotional campaign, you know, for selling this. So HTS is accepted as a term, but I don't think, because this is not the area. We had examples where lots of different terms were used in the world. So we had an issue with that. But in this case, these are basically these two, and everybody is already familiar with that.
You could also argue that already ONT and PacBio are third generation sequencing. So
well,
HTS is maybe more precise.
Just to defend NGS, I mean, yeah, you know,
don't try.
Yeah, no, but basically, I mean, of course, the next generation. I mean, it doesn't matter what the next one is; it's the next generation. So to me, you know, I mean, it the old one does. Yeah. Well, yeah, but again, you know, I mean, I think it seems to be a preference, whichever world, part of the world you're in, or whichever document you're looking at. But don't forget, some people also use the term MPS, massive parallel sequencing. That was the original term. So you might say, so yes, some of the regulators do know the different terms.
I'm not old enough for that. That's-
Oh, maybe you're too young to know that. Yes.
You didn't read the WHO TRS in that case.
And honestly, we had the question, is NGS the same as HTS? And we're happy to have this question because this is a really easy one to answer. Okay, so this is really not a challenge. You may have more challenging question to answer and to explain. This one, you will be happy to have it.
And I think when drafting the ICH document, we even tried to adhere to the HTS, but the ICH colleagues say, no, don't use HTS because it's being confused with high throughput screening. Yes, and
Other terms for HTS. Okay, so I think we've resolved that. Everybody knows the term now, the terms. Okay. I don't know, how are we doing on time? But I definitely want to get to the last question.
20 minutes.
Okay, so maybe we can you want to get to the last one or, well, can we address, I think we've addressed what are the current gaps and what are the needs. I think we have a flavor for that. Is there something additional that anyone wants to comment on? Yeah, there is.
Maybe I comment a bit on the cell therapy side because now I'm in that area. And it's very interesting because I think a lot of the company are also working on HTS and NGS, and we use whole genome sequencing for, WGS and use it slightly differently in, characterizing cell banks and maybe AAVs. So we're familiar with the technology. And at the same time, I think many of us are aware of the viral testing work that's happening in vaccine. And seeing the similarity between where the testing could happen. But I think at the same time, a lot of the regulatory bodies actually have two separate groups for the cell and gene therapy versus the vaccine, and they don't always align and talk to each other. So the challenge of alignment across different regulatory bodies and then also within the same regulatory body. So I would share, we're maybe in some ways better in that we see the ICH Q5A guidelines and the European Pharmacopoeia that's come along. So I think that's a way ahead than where we were before. And I definitely am hoping it doesn't take a milestone event to push the cell therapy into using it. But that's a challenge that I see for us as well in that space.
Maybe just to add, I think Laurent already mentioned that in the revised chapter of European Pharmacopoeia, it will be elaborated. We are also receiving a lot of questions in WHO how to deal with the evaluation of cell and gene therapy products beyond what is the application of HTS. But in many aspects, cell and gene therapy products do require special considerations, starting from the raw material, which is precious and is not available in the quantity that we have in the case of vaccines and also many other issues. So I think that we all need to think about the special considerations for cell and gene therapy products and maybe devote a bit more time in the future discussions.
No, only one point which remains a question mark regarding the evaluation by the agency corresponds to the limit of detection which is expected. Because you can validate a different limit of detection. In fact, it's your choice. But at the very end, and we begin to see some agencies that request something which is not reachable, even by PCR. So I think this is one of the questions that could correspond to a problem during the evaluation, late issue, I would say.
Well, I can just say, again, it's an evolving situation even in the agencies, and even and also sharing the align or being aligned in different offices. But I think it's good to have those discussions externally, internally. Good to see data, because I think I mean, of course, submissions with data drives the thinking forward, and we can see what do we have now, what's achievable, and again doing from the spiking studies, that's one of the goals, to see worst case scenario, different labs are using different independent protocols, if using the same sample type, what is achievable? So that sort of can also help, come up with some type of a realistic number.
Is it enough? The question is it enough?
Yeah. So like I said, I mean, it might be enough today, but it may not be enough as the technology further evaluates. And if it's enough today, then you evaluate it based on what else can be done. And do you use it to supplement or complement or, replace? So I think it's great to have these type of meetings, see data, for regulators as well as for others, that are here, to help, build the thinking as to, what the broader users can achieve. Not just one lab has got, okay, they can pick up, one virus in whatever, 10 mLs or something, but, what is achievable, as a LOD by, different labs. So the expectation, has to be more realistic as opposed, but then we also, suggest, we know that you can improve it because, we may have seen data that we know it can be improved, and then we can make suggestions how to perhaps improve it. So everybody can.
I just want to comment on that. I fully agree that we need to make progress on the sensitivity on limit of detection. However, we should keep in mind that for the existing assays, there was no target, which was, there was no target which were expressed in any regulatory documents. So we should not be more stringent with the alternative methods than we were with the previous ones. It's just to make sure that but everybody should not be more stringent, in my opinion, because in fact, you need also to consider the fact that the breadth of detection is fantastic in theory, and that the cases of contamination, as it was presented, came from the gaps of the existing methods. So do we accept the gaps, or do we try to fill in the gaps? Aligned.
The other thing, I should also say is that, the LOD may not be the same based upon the test material. If you're testing a cell bank, a one-time cell bank, versus if you're doing, at the unprocessed harvest stage where there may have been some amplification, but then there may be more inhibition. So it's not a simple answer. I think it really, when we evaluate a package, it's really in terms of the whole context of the testing and the stages being tested at, and also, if there are other processes are in place. So it's not an easy, so that's why right now we're still doing case by case, and we're hoping we can, with additional data, additional information, we can move towards having some sort of, expectation that can be generally applied.
And sorry, Karin, I just want to say that it's also the case with the existing methods, because when you have an interference, you may have to reduce the vaccine volume that you are testing. And at the end, the LOD is not so exceptional. And I am not talking about the variability we have seen yesterday on the in vitro test. In the NIMBLE study, you have also big variability in the in vitro test in cell culture. No, as well. So we have to take that into account as well.
So, and I totally agree that your evaluation is necessarily within the context of the vaccine and all the testing package that is done in parallel. And I was wondering, considering that there is no reference for the transcriptomic approach right now, did you already accept, or what will make you accept a transcriptomic approach as a release test, as a substitution of the other test? Because in supplement to the other one, I think it's easy. You're just happy to see this additional testing. Have you already accepted some product for which there was a substitution with a transcriptomic approach? And what was the criteria to say that, yes, the level of sensitivity is sufficient? And I'm not asking you, I'm asking the regulator, to be honest. Of course, you will say yes, but I want the point of view of the regulator. What are the requirements?
It was considered as sensitive enough. So the demonstration was that the level of detection was something, and it was considered that enough. In practice, you know, when you have a cell line, you do not need a very high level of sensitivity. That being said, this kind of approach is very sensitive. And to reach the level of sensitivity of such an approach, you have to dilute the cells, infected cells, down to 10 to -4, 10 to -5, etc., etc.
Mark, I would like to have the answer from a regulator.
Yeah, but I know that. But I know that. I know that. I know that. But, okay.
So we have very limited initial experience with a transcriptomic submission. Okay. And it's and again, I'm only speaking for my office. You know, the other office is also considering, but you know, in terms of the actual submission, and it's a clinical stage, early stage, and we're, you know, we evaluated it. We, you know, the whole package, if you will, and you know, we provided a lot of comments, and we're working with them on how to, you know, best use it for the suitable purpose. So we have early experience with that. So I don't think we can, you know, come up and say, well, this is the expected LOD for that. I think there's more experience with the viromics based upon all of the studies. But I should also say, you know, there are transcriptomic studies that are going on, you know, in other places. Mark presented in spiking study 4, and that actually will be a valuable study, because there you've got, you know, again, to me, that's worst case scenario. Everybody's using their own SOPs, their own pipelines, starting with the same material. So I think that information will give you, well, it might give you, this could be the, you know, the least, or you have to get at least this LOD and better. I'm not saying we're not establishing the LOD for transcriptomics here, but I'm saying that's sort of a consideration as to, you know, what can you expect at this time, and then decide, you know, is the rest of the safety package acceptable if this is the LOD you have, if it's transcriptomics, and what else can be done, you know, to test it further or to achieve better LOD. So that's how we sort of work with the sponsors. And if it's absolutely not acceptable, then it's not. But usually, you know, if it's acceptable, or if it's near, you know, then we help them, you know, what can they do to improve it. And it's perhaps that, you know, in the setting of how they're using it now, you know, the safety can still be addressed. But if they want to, you know, move further ahead, what do they need to, you know, to achieve better? So, you know, for all the NGS where we've been pretty much working hand in hand, and not just, that's the reason we don't have formal recommendations. We can't put out something that may not be achievable, you know, or maybe, you know, raising the bar too high. So we're waiting to see, you know, how to come up with something that's reasonable and confident, assuring in terms of the safety as well.
And we may have better idea in the next IABS conference that we can discuss.
Yes.
Maybe you have your point of view on the transcriptomic and no?
I cannot more agree than with what Mark said, and I said it also in my talk. Is I think that the transcriptomic approach has some conceptual attractivities. Yes, and the first thing is really we have sometimes a high abundance of transcripts, and so this and that's this predestination for testing cell culture, because you see the active replication going on, and also the diversity of the viral particles, of the genomes, of the nasty circular genomes. Also, it's much more uniform with the transcripts. So cells are easier to crack, and probably the physical nature of with the secondary structures also. So it's more uniform. So and this is attractive for broad viral detection assay.
So I just wanted to comment. So the studies that people are doing right now, they're sort of, I think, you optimized for detection, because all the cells are infected, and they're all probably expressing some level of the transcript. So but what we are concerned about is in a cell substrate, if you have a few infected cells, even if those infected cells are producing high number of transcripts, can you detect those few infected cells in the background of your cell substrate? So that's really what we are trying to, so you have to know what is the level of expression in the per cell, and then do the spiking with how many, in the background of uninfected cells. So I think most of the studies, they're with cells that are infected with the virus. All the cells are probably infected and expressing.
So you are perfectly right. There are two expression to read out the ratio of infected to non-infected cell, or a molecular number of copy of RNAs per cell. That being said, what is the level required? Whatever the readout, we don't know. So we try to be as sensitive as possible, which is easy for a transcriptomic approach because of the amplification of the signal when the virus begin to replicate. But we don't need, in reality, we would like to know what could be expected. And I know the response. It's a case by case. Yes, I know.
No. Hopefully next year we'll have a better answer with the results. I mean, to me, data speaks more than any other discussions that we can have. And I learned that very early on, so it's easier to do the study than to have year-long discussion and still not know what the answer is. So that's what we're all trying to do here. And that's the reason for the standards that we're working on, is that we're assessing the level of expression, and then based on the level of expression, then you can do the spiking with different ratios and then know what levels you can pick up, copy numbers. Yeah.
I'm just wondering about a collaborative study comparing the genomic approach with the transcriptomic approach, having an infected cell culture. And
actually, I just had a quick comment. Just to Simon's comment and about consistency of review across different, review groups within an agency. It's things like this. So when I go back, we have series of training sessions we want to have with all of the different reviewers, because I routinely will survey the different review groups to see what their experience is in NGS. And it's, it's a little bit all over the place, but for the most part, because it's very early stages, or most of these submissions are in the clinical trial stage, most of the review groups are not going into a great deal of detail for some of this. So we want to try to at least get all the assessors across every product line at least on the same page, and then at least having a few people who are knowledgeable, who people can go to consult. And that's really the only way we can do it. But until real submissions come in and we start looking at them in real detail, that's the only time you can really know whether you know what's going on or not. So
it just to be honest, when I go out here, coming back to the question here, I'm a little bit frustrated because we are discussing detection limits, etc., even though we all know the breadth of detection is superior about any of the assays. And I'm a little bit afraid, there may be a contamination case, and which will be detected by NGS, and people will ask, why was this technology not applied? And it was not because the sensitivity was not demonstrated well enough. It was just because of the breadth of detection. I'm thinking especially about cell therapy products. In the allogenic area, we're talking about cell substrates, which are the final product. When these are donor-derived material, so we are not talking about a continuous cell line or something like that. So all sensitivity we demonstrate is related to well-adapted virus strains. The Gombold paper, which was frequently commented here, they will grow up, the virus were grown up on cells which were used in the in vitro assay. Really not surprising, right? And so what I just want to say, the benefit of NGS is the breadth of detection, okay? And any sensitivity we document for different viruses, we don't know for the rest of the viruses. One have to keep that in mind. And when we go back to the in vivo assay and the vitro assay, we We validate the in vitro assay using the positive control. This is just demonstrating the performance, but it's not a validation, right? And we don't have any clue about what is the sensitivity with respect to any kind of virus which may be in the world. So I would like, to be honest, if I would be a manufacturer of a cell therapy product in an allergenic area, I would say, use it. You secure the company, you secure the people, because the risk is high, and you may even have viruses in this material, which are primary viruses, which may not adapt to in vitro assays. You will not detect them. They might be there.
So please don't get me wrong. We're not saying that we're not saying no to using NGS. Yeah, I mean, yeah, for sure. Yeah, and even in the cell and gene therapy field, I am working with the office, and they are receptive, and they are, but they haven't gotten as many submissions as we have, so they're still on a learning curve. But but they're open to discussions, and they're doing reviews and working towards it. So, again, you're hearing the people at different stages. Even within the regulators, we're at different stages, as you heard from Michael. But
no, it certainly makes a difference when it's investigating well-adapted cell cultures where you need to be afraid of some highly adapted cell culture strain, or when going more to primary materials, and certainly coming to the field of ATMPs, where a lot of primary materials are used, where a lot of blood is used, human serum, and so that there's really much more demand on the breadth of detection. And if we would go for our Mexican blood donors with a conventional 28 days in vitro testing, we would not detect the hepatitis E, we would not detect the hepatitis B virus and many viruses. So really ATMP field is completely different, and that there's much more benefit, I would say, for the breadth of detection and for the molecular assay.
But the limit of detection here should be seen as an assessment of the performance of the assay, not for a particular virus in particular. If you see that one virus is totally with a really bad LOD, then you know that you have to work a little bit on your assay and improve it. But and that's why I always comment when people use the word accuracy in parallel with HTS. We don't look for a precise LOD. We just want to make sure that we are performing the assay the best possible way with the current status. And this is where the model viruses are important, and the sensitivity is important. It's not reaching a specific LOD per virus, but it's having a pipeline that works with the current knowledge, to my view.
Yeah.
I think it's well recognized that NGS is the superior assay for breadth of detection. There's no doubt about that. And I think it's recognized that the effect of the matrix affects the sensitivity. And that's what, you have to consider in terms of, the suitability for purpose. And also, even if it's not absolutely necessary for that particular, step, you still want to work towards, conditions to optimize it. And, so that's really where I think we are. I mean, and we're working towards that too, challenging substrates. And you heard all of the talks. Some of the talks also describe the challenges in different matrices. So I think it's being evaluated. It's under evaluation, and you want to do the best you can. But yes, we have put NGS at a higher bar than PCR. That bothers me. But I think it's good because this is where we are. We expect more.
Maybe a last
Not
question.
We meaning the agency, we meaning as scientists.
Sorry to interrupt you. Maybe a last question, and then I will go to the last question for the panel discussion.
Yeah, just to piggyback a little bit on this discussion, because at least I have a little bit of history in machine learning. So I've been wondering a lot if we are overfitting to these model viruses in trying to achieve the lowest possible LOD, and then probably in our chase for that, neglecting the breadth of coverage, as was just mentioned. Is that something that others have considered? Just a general question also to the participants here, but or if it's just me.
No, when we talk about when we talk about detection, we're talking about demonstrating detection of all of the viruses and, you know, and demonstrating what is the sensitivity for all of the viruses. What's your LOD? So it's not, you know, but we do want the data for all of the different detection to, you know, the differences in the detection of the viruses also.
Yeah, that's what I mean, because you're trying to get them all on the same level. So you are,
well,
you could be overfitting your wet lab procedures to prefer these, the 5 and now the 7 viruses that you have in your, and of course I know that some of you are, and some of the participants here have had different panels, and but I've just been thinking about it, that if we are doing that.
Well, it's really to test the characteristics of the virus families that represent diverse families of viruses with different properties. So, you know, these are viruses that were selected from those families.
I know that
to challenge
the
the work.
consideration of a model is that why you're doing it, that you're trying to represent
Mm.
many, but you could do something that you're not aware of.
But the
At
idea
least
is
in machine learning, it's very easy to do.
Yes, we are never perfect. I think the model viruses have been carefully detected, and I think specifically for the vet part, I think they really represent a reasonable breadth of capsid structure, of genome structure, and so on. Of course, there might be gap with certain pre-treatments or so whatever, when you go for an ultracentrifugation, whether you can really pellet everything. So every new method might be reconsidered, and that's why I had on my slide it's a little bit variable, yes, depending. But I think for the library construction or the vet part, I'm quite confident, and also going for viral inactivation also. So when we are assessing viral inactivation, we don't want to see hundreds of viruses inactivating. Also here we are doing a careful selection of various viruses with different properties, and then we are concluding on any novel viruses. And the history, the last 30 years have shown it worked against many novel viruses. So for the vet part or for the end-to-end validation, I'm quite confident with the panel. Of course, when it comes to the breadth of the detection in the bioinformatic part, here I see the gap, and we are reflecting it when drafting. And here I would really appreciate further reflection, further work going on how to develop bioinformatics standards.
I think I could get ideal performance if I made a database of those 5 viruses and their variants, or 7 viruses and their variants. And so I think the overfitting challenge is probably okay in the wet lab, because it would be difficult, I think, to overfit, but bioinformatically, it's definitely a risk. So the idea of having challenging it with a variety of different, maybe synthetic sequences or whatever, could really help, because I do agree with you that it is a risk.
Yeah, while the microphone is going there, in terms of standard for bioinformatics, we didn't discuss that, but come to the AVD-TWG subgroup DE, this topic will be discussed next year.
I just wanted to add that last year I raised also this concern, but as the colleague said, it's not only in the laboratory. In the laboratory, I think the 7 virus panel works really nice because, as Johanna said, it's a very well distributed spectrum of the different viruses. But bioinformatics, we can have 2 type of problem. One is that someone wants to cheat it and practically do a targeted analysis just by these 7 viruses. And of course, they will have a much better sensitivity for those than the others. But could be also unintended when we start to use machine learning or artificial intelligence approach, and the system learn that we always want to catch them, then will be totally unintended, and we will have a big bias. That's why, as Johanna said, a broader panel and different panels for challenging the bioinformatics will be better.
Okay. I think we will stop here. I will just ask the last question, which is on the screen, to any of you who wants to answer. So what? Yeah, but yeah, if you want, what are the 2 important take-home messages about NGS implementation that you will share with your organization following this meeting?
So for me, from what I heard, it was calls to really to improve the regulatory acceptance and also the wider implementation of NGS. And that was not only in Europe and North America, but also beyond. And I think what we are doing is, in fact, with the regulatory standards, should play a role in that, because it's meant to support not only those who are going to implement NGS in GMP environment, but also the regulators. So in the discussions with the sponsors. So we still have some work to do at the level of the pharmacopoeia. It's not quite finished yet. We had received a large number of comments, I think around 300 to give you an idea. But it shows the interest. I think they were very positive, shows the interest, and it was also very constructive. So we still have some to address, but what we are currently hoping is to be able to present that for final adoption, possibly already in March session of the European Pharmacopoeia Commission, or maybe in June, we'll see. But possibly presented for final adoption in first half 2025. And if we are successful, then for that we need full consensus among 39 member states. So if there's one opposition, it wouldn't fly. So we need a full consensus. And then it would then be published 6 months later. And then there would be 6 further months for the actual entry into force of the document. But I think I think there is a role to play for this chapter in helping also beyond Europe. It's a good resource, I think, for both the manufacturers and the regulators.
Okay, take-home message for my institution. It's certainly also to make our institution aware that it's a coming technology that we need desperately, even at our institution, expertise. We need expertise handling big data, handling metagenomic science. We need to have the bioinformaticians and certainly thinking on the network, of course, once having it, or the role that the Paul Ehrlich wants to play also in providing training and support for us. But we need the expertise. And even in our institute, we have regulators that are colleagues that are not familiar, and we will be as overloaded when they once get to assess such product. First take-home message, us also thinking on the distribution of the standards and so how to improve it, knowing the limitation, but certainly as having the standard at different location might help at least a little bit. But I think this would also be a reflection also for our group or our consultant to reflect how to improve the standards, or whether we could go for some virus-like particles or something which would facilitate the distribution.
Thank you, Johannes.
Maybe positive message here. We saw in the history through the different meetings a lot of progress in first the technology, solving or trying to address the different challenges. It's not yet solved, and Simon summarized that at all the steps there are still challenges, and we saw a lot of people trying to solve or deal with the different challenges. We saw also a broader adoption of NGS worldwide. It was presented by Ivana. We still need to continue with maybe material or starting material for broader adoption. It's time now.
For implementation of new technology, it's very important to discuss internationally and conduct. I think multi-center study is very important. So I think that spiking study is very important, play a crucial role. And so if you are interested in the study, please join the spiking study. Yeah.
Maybe I will first let them know that it's really great to have some innovation space within a company where we can explore technology ahead of their implementation. And again, thanks to the one who push this type of evaluation really early at Sanofi, because now we have a beautiful tool in hands that require a lot of expertise, a lot of energy, and so to be able to keep this innovation space is really important. But also we need to be able to strengthen the team that is currently running this test, because it's a beautiful tool, but it relies on really fine expertise that we need to strengthen.
So very good achievements, actually. And I'd like to thank everybody who shared results and progress, and also to congratulate, because it's really fantastic to see how much has changed in the past few years since the last discussion. So I would like to wish also everybody to continue. And I think that the first take-home message is really that a lot of work needs to be done to make sure that this will be used as a regulatory tool at the global level. So building the expertise, sharing experience, and really making sure that this is accessible and well understood as a tool with a good capacity. And the second take-home message is really that number of practical issues regarding the use of the WHO panel of 7 viruses is also is an area that requires a bit more thoughts and feedback from users in order to make sure that this is broadly available, accessible, and used as intended.
And I think for me, and I agree with what's just been said, that we maybe went from, it seems like, or it looks like it can be done, to, it is being done, and people are doing it. And that's a great take-home message, I think, in the implementation and how far the group has come along. And maybe link it back to the cell therapy comment earlier, that, that was just a very general comment, but it's a way to show that group as well, and where I am as well, that, there is a path forward, and there's guidances out there now that we can follow, which is great. And so look forward towards that.
So I joined this IBS meeting since 2017, when I was not even joining our first group. And I do see the advance of this technology is quite encouraging, especially for this year. I come across all of the poster and also the presentation you have. My thinking is, regardless of the technology you use, bioinformatics are so different, and also somebody use the enrichment, somebody use some technology to further improve their sensitivity. I still have some sort of impression that things like we have the agreement of the LOD that's around some ranges, so that's really encouraging, saying that even with the different protocols, we still can reach some sort of agreement about the sensitivities. So I think that's really encouraging. And second message is not only for my own organization, but also for you, is that if you are not sure about what regulatory agency asks you to do, please do have a meeting with us, because I'm so tired of writing IR letters. So please, if you really have any concern before implementing the NGS and you really need to know what we want, please have the meeting with us before you really implementing the NGS.
Okay, so I think I would report that there is clearly an acceleration in understanding, acceptation, and that probably NGS will replace classical tests more rapidly than anticipated. And I will propose that it is time to anticipate the upscaling of our capabilities.
So for me, what I've seen is that there is a huge progress since the last meeting. There is more and more people who use NGS, and we have seen a lot of data on thresholds that we can achieve. I have a concern because I'm working in a vet vaccine, and there was very few people from the veterinary vaccine area, and we are facing a challenge. And I was very interesting to see that there was a huge progress in the database with the curation database. And interesting also to the progress that could be done for retrovirus. For us, it would be very huge point because with avian cells, we have a lot of retrovirus, so it would be very, it's an area where we can progress, I think.
I guess I can share a couple of thoughts also. So I guess, and I know many of you have been, followers of this meeting, and you may have heard this, that in the first meeting, the question was, okay, where are we with NGS? And then the second meeting, in my mind, the question was, are we getting there? And then it was, yes, we're getting there. And now it's like, we're not there yet. So, but there's hope. And I think, in putting together the meeting summaries, it really helps to see how far, where we were and how far we've come. And I know, hearing all of these challenges and issues, it's a little bit, sort of, I don't want to say depressing, but it just, burdensome is the word, that we still have a lot more to go. But I think we have to recognize how much we've achieved. And and that's really for all of you and everyone on the panel and the organizations that support these meetings and the openness that we saw in the presentations. I think that definitely is a take-home message for me that, we need to continue encouraging, these type of presentations and publications, just so we can understand better and what is needed. And also, I think it provides a resource and also, I think, confidence to those that haven't started, maybe make them a little bit less fearful of getting into NGS. And I think many of you may remember initially, the comments, there was fear, of touching it because, we know we may find something, what are we going to do? And I think, my response has always been, well, if you don't find something, somebody else will. So it's better that you find something. So, but I think with that thinking, I think many people have accepted it. They're moving along, got this far. Regulators are working hand in hand with CROs, with sponsors. And I think it's really the collaborative effort, the collaborations from everyone that has gotten us to where we are now. And I think we should all recognize that. And yes, we still have to move ahead, but we're evolving as the technology is evolving. And I think, as a regulator, I can tell you, we don't have all the answers, but we're working, trying to get them. And we don't want to put something in stone right now, and then it's not feasible or practical, and then, it needs to be changed. So it's easier to have the discussions and even do case by case, as long as, we're still moving ahead and everybody's achieving their goals. Yeah.